Hello everyone, welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of peroxisomes and lysosomes. We would also be looking at the various functions associated with peroxisomes and lysosomes. Structure and function of lysosomes. Lysosomes are membrane enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers such as proteins, nucleic acids, carbohydrates and lipids. Lysosomes function as the digestive system of the cell, serving both to degrade material taken up from outside the cell and to digest obsolete components of the cell itself. In their simplest form, lysosomes are visualized as dense spherical vacuoles, but they can display considerable variation in size and shape as a result of differences in the materials that have been taken up for digestion. Lysosomes thus represent morphologically diverse organelles defined by the common function of degrading intracellular material. Lysosomal acid hydrolases. Lysosomes contain about 50 different degradative enzymes that can hydrolyze proteins, DNA, RNA, polysaccharides and lipids. Mutations in the genes that encode these enzymes are responsible for more than 30 different human genetic diseases, which are called lysosomal storage diseases because undegraded material accumulates within the lysosomes of affected individuals. Most of these diseases result from deficiencies in single lysosomal enzymes. For example, Gaucher's disease results from a mutation in the gene that encodes a lysosomal enzyme required for the breakdown of glycolipids. An intriguing exception is the eye cell disease, which is caused by a deficiency in the enzyme that catalyzes the first step in the tagging of lysosomal enzymes with mannose-6-phosphate in the Golgi apparatus. The result is a general failure of lysosomal enzymes to be incorporated into lysosomes. All of the lysosomal enzymes are acid hydrolases, which are active at the acidic pH that is maintained within lysosomes, but not at the neutral pH characteristic of the rest of the cytoplasm. The requirement of these lysosomal hydrolases for acidic pH provides double protection against uncontrolled digestion of the contents of the cytosol. Even if the lysosomal membrane were to break down, the released acid hydrolases would be inactive at the neutral pH of the cytosol. To maintain the acidic internal pH, lysosomes must actively concentrate protons. This is accomplished by a proton pump in the lysosomal membrane, which actively transports protons into the lysosome from the cytosol. This pumping requires expenditure of energy in the form of ATP hydrolysis, since it maintains approximately 100-fold higher proton concentration inside the lysosome. Endocytosis and lysosome function. One of the major functions of lysosomes is the digestion of material taken up from outside the cell by endocytosis. However, the role of lysosomes in the digestion of material taken up by endocytosis relates not only to the function of lysosomes, but also to their formation. In particular, lysosomes are formed by the fusion of transport vesicles budded from the trans-Golgi network with endosomes, which contain molecules taken up by endocytosis at the plasma membrane. The formation of lysosomes thus represents an intersection between the secretory pathway through which lysosomal proteins are processed and the endocytic pathway through which extracellular molecules are taken up at the cell surface. Material from outside the cell is taken up in clathrin coated endocytic vesicles, which bud from the plasma membrane and then fuse with early endosomes. Membrane components are then recycled to the plasma membrane and the early endosomes gradually mature into late endosomes, which are the precursors to lysosomes. One of the important changes during endosome maturation is the lowering of the internal pH to about 5.5, which plays a key role in the delivery of lysosomal acid hydrolases from the trans-Golgi network. Acid hydrolases are targeted to lysosomes by mannose 6-phosphate residues, which are recognized by mannose 6-phosphate receptors in the trans-Golgi network and packaged into clathrin-coated vesicles. Following removal of clathrin coat, these transport vesicles fuse with late endosomes, and the acidic internal pH causes the hydrolases to dissociate from the mannose 6-phosphate receptor. The hydrolases are thus released into the lumen of the endosome, while the receptors remain in the membrane and are eventually recycled to the Golgi. Late endosomes then mature into lysosomes as they acquire a full complement of acid hydrolases which digest the molecules originally taken up by endocytosis, phagocytosis and autophagy. In addition to degrading molecules taken up by endocytosis, lysosomes digest material derived from two other routes, phagocytosis and autophagy. In phagocytosis, specialized cells such as macrophages take up and degrade large particles including bacteria, cell debris and aged cells that need to be eliminated from the body. Such large particles are taken up in phagocytic vacuoles or phagosomes, which are then fused with lysosomes, resulting in digestion of their contents. The lysosomes formed in this way can be quite large and heterogeneous, since their size and shape is determined by the content of material that is being digested. Lysosomes are also responsible for autophagy, the gradual turnover of the cell's own components. The first step of autophagy appears to be the enclosure of an organelle in membrane derived from the endoplasmic reticulum. The resulting vesicle then fuses with a lysosome and its contents are digested as described in the image. Autophagy is responsible for the gradual turnover of cytoplasmic organelles. Peroxisomes. Peroxisomes are small membrane enclosed organelles that contain enzymes involved in a variety of metabolic reactions, including several aspects of energy metabolism. Although peroxisomes are morphologically similar to lysosomes, they are assembled like mitochondria and chloroplasts from proteins that are synthesized on free ribosomes and then imported into peroxisomes as complete polypeptide chains. Although peroxisomes do not contain their own genomes, they are similar to mitochondria and chloroplasts in that they replicate by division. Functions of peroxisomes. Peroxisomes contain at least 50 different enzymes which are involved in a variety of biochemical pathways in different types of cells. Peroxisomes originally were defined as organelles that carry out oxidation reactions leading to the production of hydrogen peroxide. Because hydrogen peroxide is harmful to the cell, peroxisomes also contain the enzyme catalase, which decomposes hydrogen peroxide either by converting it to water or by using it to oxidize another organic compound. A variety of substrates are broken down by such oxidative reactions in peroxisomes, including uric acid, amino acids and fatty acids. The oxidation of fatty acids is a particularly important example, since it provides a major source of metabolic energy. In animal cells, fatty acids are oxidized in both peroxisomes and mitochondria, but in yeasts and plants, fatty acid oxidation is restricted to peroxisomes. In addition to providing a compartment for oxidation reactions, peroxisomes are involved in lipid biosynthesis. In animal cells, cholesterol and dolicol are synthesized in peroxisomes as well as in the endoplasmic reticulum. In the liver, peroxisomes are also involved in the synthesis of bile acids which are derived from cholesterol. In addition, peroxisomes contain enzymes required for the synthesis of plasmalogens, a family of phospholipids in which one of the hydrocarbon chains is joined to glycerol by an ether bond rather than an ester bond. Plasmalogens are important membrane components in some tissues, particularly heart and brain, although they are absent in others. Peroxisomes play two particularly important roles in plants. First, peroxisomes in seeds are responsible for the conversion of stored fatty acids to carbohydrates, which are critical to providing energy and raw materials for growth of the germinating plant. This occurs via a series of reactions termed the glyoxylate cycle, which is a variant of the citric acid cycle. The peroxisomes in which this takes place are sometimes called glyoxysomes. Second, peroxisomes in leaves are involved in photorespiration, which serves to metabolize a side product formed during photosynthesis. Carbon dioxide is converted to carbohydrates during photosynthesis via a series of reactions called the Calvin cycle. The first step is the addition of carbon dioxide to the 5-carbon sugar ribulose 1,5-bis-phosphate, yielding 2 molecules of 3-phosphoglycerate. However, the enzyme involved, called Rubisco, sometimes catalyzes the addition of oxygen instead of carbon dioxide, producing 1 molecule of 3-phosphoglycerate and 1 molecule of phosphoglycolate. This is a side chain reaction, and phosphoglycolate is not a useful metabolite. It is first converted to glycolate and then transferred to peroxisomes, where it is oxidized and converted to glycine. Glycine is then transferred to mitochondria, where 2 molecules of glycine are converted to 1 molecule of serine with the loss of carbon dioxide and ammonia. The serine is then returned to peroxisomes, where it is converted to glycerate. Finally, the glycerate is transferred back to chloroplast, where it re-enters the Calvin cycle. Photorespiration does not appear to be beneficial for the plant, since it is essentially the reverse of photosynthesis, that is, oxygen is consumed and carbon dioxide is released without any gain of ATP. However, the occasional utilization of oxygen in place of carbon dioxide appears to be an inherent property of Rubisco, so photorespiration is a general accompaniment of photosynthesis. Peroxisomes thus play an important role by allowing most of the carbon in glycolate to be recovered and utilized. Peroxisome assembly. As already noted, the assembly of peroxisomes is fundamentally similar to that of mitochondria and chloroplasts, rather than to that of endoplasmic reticulum, Golgi apparatus and lysosomes. Proteins destined for peroxisomes are translated on free cytosolic ribosomes and then transported into peroxisomes as complete polypeptide chains. Phospholipids are also imported to peroxisomes via phospholipid transfer proteins from their major site of synthesis in the endoplasmic reticulum. The import of proteins and phospholipids results in peroxisome growth, and new peroxisomes are then formed by division of old ones. Proteins are targeted to the interior of peroxisomes by at least 2 pathways, which are conserved from yeast to humans. Most proteins are targeted to peroxisomes by the simple amino acid sequence serine, lysine, leucine at the carboxy terminus. Other proteins are targeted by a sequence of 9 amino acids at the amino terminus, and some proteins may be targeted by alternative signals that have not yet been well defined. PTS1 and PTS2 are recognized by distinct receptors and then transferred to a translocation complex that mediates their transport across the peroxisome membrane. However, the mechanism of protein import into peroxisomes is less well characterized than the mechanisms of protein translocation across the membranes of other subcellular organelles. In contrast to the translocation of polypeptide chains across the membranes of the endoplasmic reticulum, mitochondria and chloroplasts, targeting signals are usually not cleaved during the import of proteins into peroxisomes. Cytosolic HSP70 has been implicated in protein import to peroxisomes, but the possible role of molecular chaperones within peroxisomes is unclear. Moreover, it appears that proteins can be transported into peroxisomes in at least partially folded conformations rather than as extended polypeptide chains. Some peroxisome membrane proteins are similarly synthesized on cytosolic ribosomes and targeted to the peroxisome membrane by distinct internal signals. However, other experiments suggest that some peroxisomal membrane proteins may be synthesized on membrane bound polysomes of the endoplasmic reticulum and then transported to peroxisomes, suggesting a role for the endoplasmic reticulum in peroxisome maintenance. The import of proteins into peroxisomes thus appears to have several novel features, making it an active area of investigation. Interestingly, some components of peroxisome import pathways have been identified not only as mutants of yeast but also as mutations associated with serious human diseases involving disorders of peroxisomes. In some such diseases, only a single peroxisomal enzyme is deficient. However, in other diseases resulting from defects in peroxisome function, multiple peroxisomal enzymes fail to be imported to peroxisomes, instead being localized in the cytosol. The latter group of diseases results from deficiencies in the PTS1 or PTS2 pathways responsible for peroxisomal protein import. The prototypical example is Zellweger syndrome, which is lethal within the first 10 years of life. Zellweger syndrome can result from mutations in at least 10 different genes affecting peroxisomal protein import, one of which has been identified as the gene encoding the receptor for the peroxisome targeting signal PTS1. Let us now summarize what we have learned in this session. Lysosomes are membrane enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers such as proteins, nucleic acids, carbohydrates and lipids. They contain an array of acid hydrolases that degrade proteins, nucleic acids, polysaccharides and lipids. These enzymes function specifically at the acidic pH maintained within lysosomes. Extracellular molecules taken up by endocytosis are transported to endosomes, which mature to lysosomes as lysosomal acid hydrolase are delivered from the Golgi. Lysosomes are responsible for the degradation of large particles taken up by phagocytosis and for the gradual digestion of the cell's own components by autophagy. Peroxisomes are small organelles bounded by a single membrane that contain enzymes involved in a variety of metabolic reactions including fatty acid oxidation, the glyoxylate cycle and photorespiration. Peroxisomal proteins are synthesized on free ribosomes in the cytosol and imported to peroxisomes as complete polypeptide chains. Here are a few questions for you to work out. 1. Describe the ultrastructure of a lysosome with special reference to the various enzymes it possesses. 2. Explain the various functions of peroxisomes. 3. Explain the process of autophagy and phagocytosis. 4. What are the morphological, chemical and functional similarities and differences between lysosomes and peroxisomes? 5. What causes Zellweger syndrome? Here are a few books for your reference. Biochemistry by Donald Voigt, Judith G. Voigt, John Wiley and Sons Incorporated. The Cell by Jeffrey M. Cooper, Robert E. Hausman, A Molecular Approach, 3rd edition, ASM Press. Here are a few websites for your reference. www.biology-questions-answers.com/cellstructure.html, www.ncbi.nlm.nih.gov/books/nbk9930. Thank you for watching this session. Let's meet in the next session with another topic. Till then, bye.